screen visible?
Yes, very
And
much.
am I audible?
Yeah, you are.
Excellent. Thank you so much for the opportunity. And I'm Rakesh. I'm currently a postdoc at the University of Leeds. And after 2 days of really interesting talks about agriculture crops and stress, I'm going to take you out on a field trip to forests or Amazonia to begin with, and I'll drop you back into India in the southern Indian forests, hopefully within time. So I'm not from the crop sciences. So I'm talking about the responses of trees to stress, particularly heat stress, and I study photosynthesis. Now this is the team of supervisors. I work with David Culbright and Emmanuel Gloor from the University of Leeds, and Christine Foyer, who is one of the organizers of this workshop, on understanding how the forest, particularly the Amazonian forest, is responding to high temperature stress. Now, again, the context for this is why do we need to understand trees while we are talking about crops? For a simple reason that trees survive for a longer period, and then the leaves that they have go through several years or at least seasons of temperature conditions and stress conditions. So there might be an interesting way of looking at how their mechanisms are built to respond to changes in the environment. Now, with that question, let's dive straight into the largest tropical terrestrial carbon pool on the planet, the Amazonian forest. So you see a map of Southern America and then the heat map, which shows the location of our field site over here, almost in the southern borders. And this is the hottest site in the Amazonia, and you can see the temperatures, the air temperatures in the sites are reaching levels that are up to 44 degrees. This is quite common in arid conditions, but this is tropical forest, and then these dots here represent heat wave conditions, and you can see the intensity and the number of heat waves are increasing. Now, if you see the overall temperature over these two time periods in this particular site, there's a clear sign of warming. So now we have found the hottest tropical forest site, and what we did was we measured a set of tree species. We sampled outermost sunlit branches, and we took them to the laboratory, and we treated them to different temperatures for 15 minutes. We dark adapted them, and when we did these FEFM measurement, which is quite common to the community that is listening to this. Now, to explain this particular key graph here, so the secondary y-axis is the FEFM response, and so here I have overlaid the histograms of temperatures for two different periods, and what you see here is the response for 7 different tree species, and I'm showing only the end of dry period. So this is the hottest period. So for Indian audience, this is sort of summer period for the forest here. Now, when you increase the temperature of the leaves, or rather when you heat the leaves to different higher temperatures, you see that the FEFM response will remain constant, but at a certain point it declines down. So this is your typical dose response curve. And what I have plotted here, this is actually a tiny box plot horizontally, if you like. And those are the box plot for the temperature at which FEFM starts to decline for these 7 tree species. And what you see is in the recent period, those tree species are experiencing temperatures that affect their photosystem to functioning. So this is a published piece of work. This was the first work from my PhD. The second bit of work that we did in the same site is we measured gas exchange temperature response, so CO2 assimilation rate temperature response. Now, what we are going to see next are parameters extracted from this typical curve. This is for one of the tree species measured in the forest. So with increasing leaf temperature, the CO2 assimilation rates increase up to an optima, and then they go down further on. So the highest rate that we measured is referred as P-opt, and the thermal optima is referred as T-opt, right? And how do these, let's look at how do these differ across 2 seasons in this particular site. So we measured it during the end of dry period and end of wet period. So in Amazonia, there is 6 months of continuous rain in this particular site. And after about 5-6 months of dry period, we measured it again towards the end of dry period, which is the hottest period. So the hottest period is referred as the dry period here, and the end of wet period is referred as wet period. So blue is the color. Now this is all the species put together, and that's the p-opt here. What you see is typically, as you would imagine, the p-opt during the end of wet period is higher than the end of dry period, and that's quite obvious. But what is different here in the response is this one particular species, Hymenaea coberil, which is an emergent tree in this particular habitat, has higher photosynthetic rate during the end of dry period rather than the end of wet period. So this is a very distinct response. Whereas in the previous FEFM temperature response measurements, we found a species Amayoua guianensis, which showed almost 50-degree temperature up to which it could survive FEFM response. And while the same species did not have very high CO2 assimilation rate thermal optima. Now, if we compare this response for all the data that exists for the tropical forest, this is all the data that exists for the tropical forest, at least the one that has been published. So let me explain this a bit more, because I've pulled a lot of different things here to get a comparison. So these 2 sites are the Central American site from the Smithsonian Tropical Research Institute, Panama. And when they say wet and dry, this is wet forest and dry forest. There's a difference in the total rainfall that they receive, and they were both measured during the wet period typically. Whereas what you see here is our site that we measured, the hottest Amazonian forest site, and we measured during the wet period and dry period. These are not different sites. So when we say wet forest here, these are 2 different sites. That's what I want to highlight here. And then there's yet another study in Manaus where the forest is aseasonal. So here what I want to highlight is within the larger Amazonian forest biome, you will find a whole range of distribution of temperature and rainfall across geography. While this particular site, Manaus, is aseasonal, that means it receives constant rainfall for all the 12 months, and the temperatures are almost constant. Whereas Nova Xavantina, in contrast, it experiences a 6-month-long wet period, continuous rains, and then 6-month-long dry period with almost no rainfall. But that period has high temperatures, and there's a stark temperature difference. Now with that, so these 3 are the sites for which we can compare across different sites. And what you see is during the dry period, the hottest Amazonian forest site has the lowest photosynthetic rate at the thermal optima. And that is the curve that you see. This is all the species, these respective number of species pooled together into typical temperature response curves for the ecosystems here. And the dotted lines are extrapolations, the solid lines are actual data sets. While you see that these are comparable and there is different amplitude for these peaks, during the dry period, really they are experiencing stress, and then they don't see an optimal behavior, they just decline. So this is possibly the period when they're experiencing the maximum stress to their physiological functioning. Now, we would intuitively think of the question, would FEFM response metrics and the temperature response curves of CO2 assimilation rate are related? So from the limited set of species that we have, we did not find a relationship, but that's just a minor point. Now let me take you back to the work that we did during the corona pandemic. So this work was done in our India site in the Western Ghats forest. So for the ones who are not familiar with the typical Western Ghats landscape, this is again a starkly seasonal forest with undulating terrain like this. This is a typical central Western Ghats where you see in the valleys there are areca forests or in the lack of these, you'll find swamps. But the areas that are exposed to excessive wind and landslides, they are drier and they're more covered by sparsely distributed deciduous species, whereas in the valleys, towards the valleys, you'll find more of evergreen species. Now, this is the site where we have measured. So we measured 9 different tree species along this slope gradient of about 100 meters. And what you see is these 3 species, Terminalia, 2 Terminalias, and 1 Carya species here characterize the hilltop, whereas along the slopes you'll find 3 other species, which are Memisilon, Canthium, and Olea. And in the valleys you'll find much more evergreen Nema and Hopia and Garcinia, very slow growing tree species. Now, in these sites, we wanted to measure temperature response of CO2 simulation rates, but what we were worried about was when we typically measure temperature response curves on trees, we have to excise and cut the branches and then bring them down and then measure it using our instrument. And different species respond differently to that excision due to the loss of the flow of water that goes through the sampler essentially. So there is always that artifact when we are interpreting the data. But in this particular forest, due to this slope gradient, very steep slope, we were actually able to access the leaves on the tree right there without having to cut them. So the data I'm going to show you next are probably the only second set of data for in situ measurements without cutting the branches. So the other one is from the Smithsonian Tropical Research Institute, Panama, by Martin Slot, where they used canopy access cranes to literally go over the trees and measure it on top of the branches. Excuse me. So now let me just walk you through these panels here. So the top 3 species are the hilltop species. I've arranged them in the same order of the schematic that I just showed you before. And then these 3 species are the slope species, and these 3 are the species that are in the lower valley side. And again, there are these, we measured it during 2 periods. So one is the end of wet period, or November, December period in India, where there is water rich conditions. So this site gets about 4,000 mm rainfall, and then right after the rainfall there is enough water in the soil for trees to photosynthesize, and you would expect that they would be very happily photosynthesizing at higher rates. Whereas during summer there will be less water in the ground, and there will be higher temperatures. So we wanted to see what happens there. Now you see a different set of behavior. For the valley species, there's no seasonal difference, so to speak, here. Whereas for the species in the slope, these are evergreen species again. These evergreen species photosynthesize at a higher rate during summer period. Whereas for the hilltop species on the contrary photosynthesize at a higher rate during the post-monsoon period, which is water rich. So intuitively, if you think of explaining this, it just probably we can describe it that the deciduous species, which are not so deeply rooted, are able to use the water during the post-monsoon period more efficiently than these evergreen species, and able to photosynthesize by losing a lot of water. Whereas during summer they don't have so much access to water, and then their photosynthesis is reduced. While this is something which is not very easy to explain, because along the slopes there is enough water relatively, although we have not measured the soil moisture condition. That's something I want to mention here. These 6 evergreen species at least show 2 different responses. But overall, if we look at the p-opt, it did follow this, the stratification of those 3 strata very well during the post-monsoon period, during the water-rich period. Whereas during summer, the deciduous were much less in their photosynthetic capacity at the thermal optima. Now, if we contrast it against the transpiration rates that were measured simultaneously. So this is the same panel, while here on the right hand side, I'm showing transpiration rates. And what you see here is again a set of very different responses. While for these valley species, there is no difference in the transpiration rates. For the slope species, there are two types of responses, and the direction of responses is slightly different. Canthium is behaving very differently from the other species. We are not able to explain this from any of the leaf traits that we try to test it against. The most interesting ones are these deciduous hilltop species. While in some species they do follow this increase with increase in transpiration with temperature, and there's a seasonal difference. In this particular species, there's a complete decoupling. While if you map this onto each other, with increase in temperature, CO2 assimilation decreases, whereas the transpiration rates continue to increase in the post-monsoon or the wet period. So there's a lot of different stomatal dynamics happening here. We fitted, Belinda Medlin's model for these, and then there again we get different prediction power of that model. Some of the species responded quite well, whereas some species the model is not able to predict what the species is doing in the field itself. So this is to sum up. Again, this includes the data for 3 species per strata, while during the end of wet period, during the water rich period, there is this clear relationship along this strata in this forest. During the summer period, the hilltops actually tend to have much lower photosynthesis rate and transpiration rate, probably because they don't have access to water in there. So this is what I wanted to highlight. So there's a whole different set of responses in leaf temperature regulation in tree species and how they respond to temperature. I think this is something that links to what Dr. Siddiqui presented yesterday in one of his talks, while he was talking about the wheat genotypes. While he was showing differences along the genotypes, so here we are talking about different species in tropics and how they're responding. Thank you very much.
Yeah, thanks Rakesh for exhaustive and elaborative talk. We have one question in the chat box. The question is, how will you disentangle the effect of altitude against species responses in photosynthesis?
Good question. So the thing is, we don't have enough power to disentangle this. While we're trying to understand this particular forest site and then account for over 70% of the total biomass cover in that forest to represent that site, we happen to stratify these along these 3 different classifications. But if you want to say anything about elevation, I think some of the studies by Kenneth Feely and group, they studied FEFM temperature responses along the almost 2,000 meter gradient in the Andes forest. I think that is more powerful to understand those. And they did not find any relationship in FEFM temperature responses or T50s. So I don't know if that answered your question.
I think we have. And thanks, Rakesh, for a nice talk.
Thank you so
much. All the best for the future research.
Thank you.
So with this, we end today's session. On behalf, I thank all the speakers and also the organizer for giving this chance to the chair and Florian. You want to add some sentence to them or some remarks?
Yeah, I just want to agree with you. Thanks to all the speakers, and good luck with your future work.
So thank you very much, all the speakers, Gaurav, Bala, Manjari, Vipasha, Ashish and Rakesh. It has been a wonderful evening we spent together. We learned a lot. Despite time provided was very less, I understand that everybody must have felt that 10 minutes is not good enough, but that's the challenge for the youngster to be able to present their work in 10 minutes. I think the session has been wonderful, and thanks to both the chairs, Supriya and Florian. We enjoyed yours. Look forward to have more interaction from these young people and would like to listen extended talks in future. I would like to say that when we have physical meet between Indo-UK collaborations later on. Thank you very much. I think we are done with today's talk, unless Christine wants to add something. Christine has been sitting silently. I would like to ask if she has to share something.
No, I've very little to add. It's been a really interesting day. So much data, so much to think about, actually. And I know we were having discussions here over lunch, talking about some of the things we heard this morning, and I assume that you will too. There are many, many interesting things that we need to think about and get in contact with the people who gave the talks in the future, you know, to go further with collaborations. It's been a very, very interesting day, and I greatly admire the amount of work that all the young people in particular have done. Huge amounts of work. So thank you very much.